Hello everyone, welcome back again. So today I have come up with a detailed explanation of CSIR Net Life Sciences Answer Key 2022, where I am going to discuss all questions from Unit 4, that is Cell Communication and Signaling. So here I am going to discuss morning shift question paper and we'll see all Part B and Part C questions of CSIR Net Life Sciences. So let's get started with the session. Coming to the Part B questions first. So the first question that was asked in Part B is about which one of the following combinations of CD molecules and their associated functions is matched incorrectly. So here you have to select the incorrect option. So here if you see, they are asking about CD. So what is CD? CD are nothing but they are cluster of differentiation molecule that are specific to different cells. So the first option is about CD1. So CD1 they have given it is an antigen presenting proteins that present antigenic peptide to T cell receptor on natural killer cell. And if you see, CD1 is a human cell surface antigen that is presented on the surface of thymocytes, or you can say T cell, or certain neoplasm on T cell. So this statement is incorrect because it is asking about CD1 and CD1 is presented on the surface of neoplasm of T cell or T lymphocyte. But here it is given about natural killer T cell. So this option is matched incorrectly. And here if you see the question, we have to select the incorrect option only. So here we have come up to the option, that is option first is incorrect. Now let's see the next option or all three options whether they are correct or incorrect. So CD8. CD8 is a marker that is present on the surface of TC cell. Now you all know that there are two types of T cell, TH cell that is T helper cell and TC cell that is T cytotoxic cell. T helper cell has CD4 marker and TC cell has CD8 marker. So CD8 is a thymic differentiation marker of T cell, that is a correctly matched option. Now let's come to third option, that is about CD11A. Now if you see CD11A, that is cluster of differentiation 11A, is a pan-leukocyte marker that is present in B lymphocyte, T lymphocyte, neutrophil, basophil, eosinophil, monocytes as well as macrophages. And what's the function of this CD11A? It is used for the cell-to-cell adhesion by interaction with ICAM1 as well as LFA. Now if you see the diagram, this is about the interaction between the antigen presenting cell and T cell. As you all know that antigen presenting cell ka function is to process and present the antigen to T lymphocytes. Now this ICAM, you can see here, ICAM is a structure that is present on antigen presenting cell. And what are the examples of antigen presenting cell? It can be any B cell, it can be dendritic cell, it can be macrophages. You can see it is presenting the antigen to T cell. But what are the interactions that are present between APCs and T cell? They are ICAM along with LFA1. Means ICAM is used for cell-to-cell adhesion, means between APC and T cell adhesion. So let's come back to the C statement and see whether it is correct or incorrect. So CD11A, which is a pan-leukocyte marker, it is a membrane glycoprotein that provides cell-to-cell adhesion. Yes, correct. By the interaction with ICAM1. Very correct statement. CD14, it activates innate immune responses by transferring LPS or LBP complex to TLR4. Now we know that there are two types of immune system, innate immune system as well as adaptive immune system. But how actually innate immune system works? So innate immune system has TLR, that is toll-like receptor, that is going to identify all pathogen associated molecular patterns. So out of all these PAMP, one is lipopolysaccharide, that is a characteristic feature of, you can say, gram-negative bacteria. So once the gram-negative bacteria enters inside the cell, that is identified by TLR4 of innate immune system. And how LPS ka binding to TLR4 is happening? It is because of the CD14 as well as lipopolysaccharide binding protein, that is LBP. Now again we'll read the statement. It activates the innate immune responses, yes, by transferring the LPS LBP complex to TLR4. This is very correct statement, and here we have to just select the incorrect statement, and we have seen CD1 that is present on T lymphocytes, or you can say thymocytes or neoplasm of T cells. So that is a incorrect option. So according to the question, the right answer is option 1, that is option A. Now let's come to the next question, that is about which one of the following is a small sulfated peptide that is secreted by a rice pathogenic bacterium Xanthomonas oryzae to modulate motility, biofilm formation and virulence. Now if you see, question has given you many hints like pathogenic bacterium means a bacterium that actually induces diseases, but it is a rice pathogenic bacterium means it is causing disease in rice plant. So what kind of disease it cause? It cause bacterial blight of rice in grasses and also in sedges. It is a small sulfated peptide. So this is a direct question that was asked this time in immunology, or you can say in CSIR unit 4. But this protein that is secreted by this pathogenic bacterium, it has the ability to modulate its mobility and also biofilm formation. And if you see, this biofilm formation is associated with virulence, and that produce, or you can say that enhances the virulent property of this bacterium. So what is the name of this protein? So this is just a direct question, and the name of this protein is AX21. What about coronatin? Coronatin is just used for the, you can say it is a toxin that is produced by Pseudomonas, and it is just used by stomatal opening and closing. So this is not produced by Xanthomonas oryzae, but this is produced by Pseudomonas species. What about N-acyl homoserine lactose? So they are not synthesized by Xanthomonas oryzae. They are used for some other processes, not here. So AX21 is only the small sulfated peptide and also not exopolysaccharide. So from here, the right option is answer 3rd. So AX21 is only the right answer and is only the peptide that is secreted by Xanthomonas oryzae. Now let's come to the 3rd question that is asked in part B section. Which one of the following small molecule or neurotransmitter is not synthesized from tyrosine? So here you have to correctly see the question, what it is asking about. So it is asking about neurotransmitter that is not synthesized from tyrosine. Means you know that neurotransmitter, they are synthesized from amino acid, whether it is tyrosine, tryptophan or any kind of glutamate amino acid. So all these amino acids are responsible for the synthesis of specific neurotransmitter. So here it is asking about which of the neurotransmitter it is not synthesized from tyrosine amino acid. So let's come to the diagram and understand the metabolism of tyrosine amino acid. So if you see the metabolism of tyrosine amino acid, you can see here the structure of tyrosine. That is, it is an amino acid. When it is acted upon by tyrosine hydroxylase enzyme, means hydroxylation reaction is taking place, and this tyrosine is metabolized into dopa, which is a precursor of dopamine. And you can see the first neurotransmitter that is formed from tyrosine is dopamine. So now we can say that dopamine is formed from tyrosine. But what happens if dopamine is metabolized in the presence of dopamine beta hydroxylase? Means second hydroxylation reaction is taking place and it is resulting to the formation of norepinephrine. And this norepinephrine, when again undergoes a single reaction, that is phenylethanolamine and methyl transferase, means methyl group is transferred and this norepinephrine is converted to epinephrine. Means overall, if we see, this single tyrosine and amino acid is responsible for the synthesis of 3 different neurotransmitters, that is dopamine, norepinephrine as well as epinephrine. So let's come back to the question and see what it is asking about. So here it is asking about which of the following small molecule that is not synthesized from tyrosine. So is it epinephrine? No, we have seen epinephrine is derived from tyrosine itself. No, not correct. Dopamine, no, it is also derived from tyrosine. Serotonin, no, we have not seen serotonin. So serotonin is a neurotransmitter that is derived from another essential amino acid that is called as tryptophan, and this serotonin is active in nerve terminus. So this option is the right answer because this serotonin neurotransmitter, it is not synthesized from tyrosine amino acid, and norepinephrine also we have seen it is synthesized from tyrosine as well. So let's come to the next question, that is question number 4. So while studying pathogenic bacteria, a protein with the following features was identified. Now they have given experiment to you where the group of you can say lab members, they were studying pathogenic bacteria. They were studying about pathogenic bacteria, and where they have found a pathogenic bacteria, one simple protein that was having following features. Now let's read out the feature and come to the final conclusion. So the first feature that is it has given, it was secreted during infection condition. So they were studying pathogenic bacteria. So it is given it was secreted during infection condition but not in vitro culture. And if we see the option an endotoxin, it is secreted during infection condition because endotoxin is produced by a bacteria itself. So when the bacteria dies, this endotoxin that is produced by bacteria, it will come out. So it is a secreted part of bacteria and it is produced during infection. Superantigen also, when there will be any kind of serious illness, superantigen will be there. Pore forming toxin means toxin that is produced by bacteria. Means if any kind of infection will be there, it will be produced by bacteria that will induces pore formation in cell membrane. So we can just make it out like this infection condition also secretes some pore forming toxin. Means toxin is secreted whether it is forming pore or not. But toxin is secreted means it is secreted during infection condition. And also AB toxin. So where AB toxin is used, AB toxin is responsible for causing cholera and pertussis, all these types of diphtheria diseases. So it was secreted during infection conditions. So we cannot make any option as a right answer through this statement. So let's come to the next statement. It was also observed, and the second feature that they observed was it is present in the membranous fraction in traces. In traces means it is present in very small amount. And if you see, this endotoxin is a part of bacterial membrane because this is a secreted part of bacteria, but when it will come out only upon the lysis of a bacterial cell. Means this endotoxin is produced in more amount in bacteria but not in traces. So this option goes wrong here. Endotoxin, they are always produced in more amount, not in traces, which was released upon bacterial lysis. So this statement is correct, but here if it mentioned more amount, so it would be endotoxin. So from the second statement, we can just make it out that endotoxin option is incorrect. Now let's come to the third statement, that is C. It had an heat labile N-terminal enzymatic domain. Okay, it's just an info about all these options that binds MSC molecule. So this is the hint that they have given in this statement. MSC molecule, if you see the AB toxin action, it doesn't involve any kind of MSC molecule. It just causes cholera and diphtheria kind of diseases where AB toxin, say A part is released and it enters into the cell. And also pore forming toxin, it doesn't bind to MSC. But if we see super antigen, so they belongs to the class of antigen that non-specifically activate T cell. Now, if they are activating T cell, means T cell activation happens by the APC, that is antigen presenting cell, because just now we have seen in the previous question, like APC was presenting peptides to T cell. And with the help of these T cells, you can see T cell ka receptor will be present. You can see this is a diagram for super antigen. This is the T cell receptor and this is MSC second. MSC second means it is presenting the peptide to T helper cell, because TC cell, it recognizes peptide when it is bound to MSC first. And if it is T helper cell, it will always recognizes peptide when it will bound to MSC second. Means T cell is only bound to MSC second. So super antigen will bind to these MSC molecule. So let's come back. So now we have omitted first option, that is endotoxin. So AB toxin, again, it doesn't bind to MSC. So again, this fourth option is incorrect. Pore forming toxin, it is just secreted by a bacteria, or you can say any kind of pathogenic bacteria, and it will just induces the pore formation. It has nothing to do with MSC molecule. But if you see super antigen, they always causes the non-specific activation of T cell by binding to MSC. And with this, you can make the second option, or you can say the super antigen option, as the right answer. So it had a heat labile N-terminal enzymatic domain that binds MSC molecules, yes, and stimulate T cells non-specifically. And if you are not knowing about anything, but if you are knowing that super antigen causes the non-specific activation of T cell, so from here also you can select the option like super antigen. Now here you can see super antigen ke liye they have given two features of their structure. It had a heat labile fragment, and this heat labile fragment is called as N-terminal, and C-terminal is a heat stable fragment. N-terminal is heat labile and C-terminal is heat stable, and N-terminal has an enzymatic property, whereas C-terminal doesn't have enzymatic property. So with this we have seen the second option is the right answer for this question. So now we have just seen 4 questions of part B that was asked in unit 4, cell communication and cell signaling. Now let's come to part C questions, and we will discuss all part C questions that are asked in this unit. The immune system of thymectomized and lethally irradiated A and B F1 mice. Means they are talking about the F1 progeny that is derived from mice A and mice B. As you all know that when mice A and mice B means they are parental species. When there will be a cross between mice A and mice B, they will form F1 progeny. So this F1 progeny they have mentioned A into B because there was a cross between A and B parents. And the genotype of this mice is H2A by B. So what is the genotype? It is H2A by B. Were reconstituted by grafting thymus from strain B mice. Now they have reconstituted this mice from the strain B mice. So now they have taken strain B mice. So they have specifically taken a strain B mice that has a phenotype, or you can say H2B. And with A into B F1 bone marrow cells. So now they have taken bone marrow cells from F1 progeny that is derived from A into B parents, and they have reconstituted thymus from strain B mice. So they have just taken suppose this is strain B mice. So they have just taken thymus from here and they have reconstituted and recultured with bone marrow cells of A into B mice, that is F1 progeny. The mice were then infected with lymphocytic choriomeningitis virus, that is LCMV. Now the reconstituted mice that has a thymus from the strain B mice. So this was F1 progeny and the thymus was grafted from strain B. Now this mice that is reconstituted, it was now infected with LCMV virus. Means this is a kind of virus that is infecting this mice. The CD8 cytotoxic T lymphocyte from the reconstituted mice were then co-cultured. So what they did after this, when they infected these mice from LCMV virus, after that they isolated CD8 cytotoxic T lymphocyte. They isolated CD8 cytotoxic T lymphocyte. Now you know that CD8 T lymphocyte, they have one function for killing the infected cell. So they have isolated CD8 T cell, means they will kill one cell. Now let's understand the question and see the statement that they have given. Uninfected fibroblast cell from A into B F1 mouse. So they have isolated uninfected fibroblast cell from A into B F1 mouse. So from F1 mouse, they have isolated uninfected fibroblast cell. Okay, from B statement, uninfected fibroblast cell from strain B mouse, uninfected fibroblast cell from strain B mouse. So now like this, they have given 6 statements. So here we have to identify which of the options below indicate the cells that will be lysed by CD8 cytotoxic T lymphocyte from the reconstituted mice. Wow. So they have just reconstituted this mice and after that they have isolated CD8 T lymphocyte. Now they are asking about the specificity of this T lymphocyte. Now you know that this T lymphocyte are isolated from reconstituted mice. But this reconstitution was done when the thymus from strain B mice was grafted into F1 progeny. Means this T lymphocyte will only have the ability to identify its own strain, that is strain B. Because from here, the thymus was grafted into F1 progeny. And you know that thymus maturation may all T cells and you can say T lymphocytes that are matured and produced in thymus itself, that are matured in thymus and produced in bone marrow. So you can see if T lymphocytes are produced and they are isolated, means thymus is grafted into F1. So they will be able to recognize the strain B mice because from there only thymus is grafted, and now it is grafted into F1 progeny that is reconstituted from A into B. So here also you can see it is grafted, so it is able to recognize the infected cells of F1 progeny as well as strain B mice, not strain A mice, because from here no thymus is grafted. So now let's read the statement. Uninfected fibroblast cells. Now just now I mentioned CD8 T lymphocyte, it is going to kill infected cells only. So this CD8 T lymphocyte, it will not recognize any uninfected cell, because if there is no infection, this will not be killed by CD8 T lymphocyte. Again, uninfection is there, uninfected cell. Now let's come to the D statement. So from this, A, B, C statement are omitted. So here also A statement is there and here B statement is there. Now only two options are left. LCMV infected fibroblast cell from A into B F1 mice. So if A and B F1 mice is infected with any kind of virus, so that will be recognized by CD8 T lymphocyte. Why? Because this is a F1 mouse. And in this mouse only we have done reconstitution. We have taken thymus from strain B and we have put it in A into B F1 progeny. So obviously it is going to recognize this infection. So yes, it is going to kill this infected cell. LCMV infected fibroblast cells from strain A mouse. No, it will not be able to recognize strain A mouse. Why? Because strain A mouse doesn't have to do with this T lymphocyte, because it was isolated and grafting was done from strain B. So it will not recognize this infection. LCMV infected fibroblast cells from strain B mice. Yes, it is going to identify this strain B infection. So accordingly, D and F statement are correct. So accordingly, option third is the right answer. So now let's come to the next question, that is about cell signaling, and it is about GPCR. The signal transduction pathway involved in glycogen metabolism triggered in the liver by the hormone epinephrine involves the following step. Now they are talking about the epinephrine growth factor, and you know epinephrine is a hormone that is inducing the GPCR pathway. Now let's read the statement and understand the correct sequential order of this GPCR pathway. The first statement that they have given activation of G protein by the activated receptor. Yes, very true. So whenever a ligand, so they have given about epinephrine. So whenever epinephrine binds to its receptor, that is specific receptor, it will activate GPCR pathway. And once the receptor is activated, and you know this GPCR pathway and receptor activation, it leads to the activation of G protein. And this G protein is a trimeric protein that is bound to GTP. So G alpha particle, G protein trimeric. Why? Because it is made up of G alpha, beta and gamma. So let's understand this with the help of diagram. So here you can see adrenaline. So here you can just imagine epinephrine hormone that is given in the question. Once it binds to the GPCR receptor, that is a serpentine receptor 7, or you can say multipass receptor, it is activated. So now this GPCR is activated. It will activate G protein that is ultimately going to activate adenylyl cyclase enzyme. And once this enzyme is activated, it will act on its substrate. And what is the substrate? It is ATP. And this cyclase enzyme will convert this ATP into the cyclic form. Name only suggests cyclase enzyme. Means it will convert this ATP into the cyclic form, and this cyclic form is called as AMP. So this ATP is converted into cyclic AMP. And once cyclic AMP is formed, it will lead to the activation of protein kinase A, and the downstream signaling events will occur. So let's come back and read the statement. So activation of G protein by the activated receptor. Yes, this is the first step. Why? Because here we have to select which one of the following combination describe these process in the right order. So we have to select the correct sequence of this GPCR pathway that is taking place with the help of epinephrine hormone. So there will be activation of G protein. Yes, correct. Protein kinase A activation. You have seen protein kinase A is activated at the last. Why? Because CAMP that is formed, that only activates the protein kinase A pathway. So where you can see the B option at the last. So here you can see B option at the last only in 2 option, and we can make this option as a wrong option. Secondary messenger generate 3-5- cyclic AMP. Secondary messenger, what do you mean by secondary messenger? Secondary messenger that is produced as a result of metabolic pathway. You can say any kind of signaling event. So this cyclic AMP that is formed, it is a secondary messenger. Adenylyl cyclase activation. So firstly, there is an activation of G protein. When an epinephrine binds to receptor, there will be activation of G protein that is going to activate a specific enzyme, that is adenyl cyclase. So this is second statement. This adenyl cyclase causes the cleavage of ATP. It will act on its substrate ATP and converts this ATP into cyclic AMP. So cyclic AMP generation, and the last is the protein kinase A activation. So the right sequence is A, D, C, B. So where you can find A, D, C, B, we can find A, D, C, B option only in the first option, that is number 1. So from here, this option goes right. Now let's come to the next question. So here you can see match the following question. So this question is very easy as it is about cancer origin. So here you have to select the tumor types. They have given you the definition and you have to select the nomenclature for that. So let's see. A tumor that has arisen from endodermal origin. And if you see epithelial endodermal origin, this tissue is called as carcinoma. This tissue is called as carcinoma. So where you can see option A with second. You can see option A with second only in the one option, that is option 2. Now let's check another options whether they are correct or not. So cancer cells that are arisen from hematopoietic stem cells that do not grow as solid tumor. And if you see hematopoietic stem cell ka cancer, it is associated with leukemia because hematopoietic stem cell that leads to the formation of all different type of blood cell. But now there is a cancer in hematopoietic stem cell itself. Hematopoietic stem cell. Now this hematopoietic stem cell itself is mutated, means it will lead to all different type of blood cell ka cancer, that is leukemia. So it matches with B, matches with 4. Where you can see B with 4 again in second option. So again we have made first option A with second, B with 4. So the right answer is option 2. A permanent change in the genome of a cell that results in abnormal growth. So whenever there will be a normal cell, upon an event of transformation, that normal cell is converted to cancerous cell, and that is why it is called as transformation. Means one type of cell is converted to another cell. Now if you see this definition is for transformation. So where you can find transformation word? You can see transformation word in the last option, that is 6. So C matches with 6. A tumor that has arisen from mesodermal connective tissue. And this is very simple. You all know that cancer that arises from the mutation, or you can say any kind of mesodermal connective tissue. So it is associated with sarcoma. It is called as sarcoma cancer. So the right answer for D is option 3rd. So accordingly, we have seen option 2 is the right answer. Now let's come to the next question. That is about cancer, and it is about viruses as well. So now let's read out the statement. Is a combination based question where you have to select the combination of a statement, whether it is incorrect or correct. So following statement are made about some of the abnormally expressing protein in human cancer. So it is talking about human cancer. So let's read the statements. So the first statement is talking about increased telomerase expression always contributes to increased cell death in cancer cell. First of all, it has the term always. So it has absolute term, but still we'll prove it how it is incorrect. Whenever there will be an absolute term, most of the time they are incorrect. But you all know telomerase expression, it is associated with cancer. How? Because telomerase enzyme, it is responsible for long-term cell division. And when long-term cell division will be there, obviously there will be a cancer. but not always, because it has seen that according to the recent, you can say the paper that is published in NCBI or PubMed, according to that, telomerase expression contributes 85 to 95% of the cancer. Not all the time. As we know that telomerase is responsible for long-term division that will turn the normal cell into a cancerous cell, but not always, because telomerase is associated with only 85 to 95% of the human cancer. So this option is incorrect. And what we have to select, which of the following statement are correct? So we have seen the first option is incorrect. So here we can just make it out a wrong answer, because here it is including option A, but we have seen that statement is incorrect. Again, option 2 will go. Now let's read the next statement. Overproduction of anti-apoptotic protein, that is BCL2, can lead to inappropriate cell survival and is associated with the chronic lymphoblastic leukemia. Now you all know that there are two types of apoptotic protein. The first is pro-apoptotic protein that promotes the process of apoptosis. Pro-apoptotic protein means precursor of apoptosis, the pro-apoptotic protein. And the next is anti-apoptotic protein. And you know that anti means against. That will stop the process of apoptosis and it will stimulate the, or you can say enhance the process of cell survival, because it is stopping apoptosis means it is enhancing cell survival. So what are the examples of anti-apoptotic protein? It is BCL2 family of protein. And what are the examples of pro-apoptotic protein? They are BAX, BAC, BID, BIM, BIC. Many proteins are there. So overproduction of anti-apoptotic protein. If anti-apoptotic protein, if they are overproduced, it will lead to inappropriate cell survival. Yes, because they are stopping the process of apoptosis and it is associated with leukemia and lymphoma type of cancer. That is what called as chronic lymphoblastic leukemia. So this answer is the correct statement. So where you can find option B? We can find option B only in one option, that is option 3. But still we'll check other statement if they are right or wrong. The E5, E6, E proteins encoded by human papillomavirus are tumor suppressor. Now you all know that human papillomavirus, they are associated with cervical cancer, and also sometimes they will cause genital cancers in male as well as female. But they encode two main type of proteins, that are E6 and E7. E6 protein is known to sequester p53 gene, which is a type of tumor suppressor gene. And E7 is known to sequester retinoblastoma gene. So P53 and retinoblastoma, they both are tumor suppressor genes that are very essential, or you can say very important for the normal functioning of cell. Why? Because they are tumor suppressor gene. They are suppressing the formation of any tumor. But if there is a mutation in both of these genes, this tumor suppression will not take place and it will lead to cancer. So if human papilloma virus, it encodes E6 protein, it is going to sequester P53. Means all these functions are lost for P53 gene, means P53 will not show any tumor suppression property. And if E7 is released by human papilloma virus, so it is going to inhibit retinoblastoma gene. Again, this is a type of tumor suppressor gene, means it will lead to cancer pathway. But if you see E5 protein, so E5 protein is associated with cell survival. It is not associated with, you can say, any kind of inhibition to tumor suppressor gene. So this protein is associated with cell survival. So it is asking about E5, E6, E7. All proteins that are encoded by HPV, that is human papilloma virus, are tumor suppressors. No, only E6 and E7 are tumor suppressors, as they are suppressing P53 and retinoblastoma respectively. So from here, this option goes wrong. And we have seen the correct option is option D. So again, this option goes wrong. So now we have ultimately reached to the right answer, that is option 3. Still we'll see the last statement. Overexpression of cyclin D. You know, cyclin D is responsible for the maintenance of any cell in the G1 phase, or loss of P16 or retinoblastoma. So whenever this cyclin D1 is expressed, it will always cause the phosphorylation of RB. This is a RB protein that is responsible for the tumor suppression. So we have seen it is a tumor suppressor gene. So cyclin D1, whenever it is overactivated or whenever this is expressed in cell, this will add phosphate group to the RB gene. And now this becomes phosphorylated, means this is inactive. When this is inactive, it will cause the expression of genes that are involved in cell survival and signaling. And when this cyclin D is overexpressed, it is always inhibited by P16 protein. But if you see, if RB and P16 loss of function will be there. So if P16 and RB genes are mutated, means overexpression of cyclin D1 will be there, because there is no one to control this cyclin D1 protein. And we have seen if this cyclin D1 are overexpressed in G1 phase, they are inhibited by P16 set of protein. This is a specific protein that is inhibiting cyclin D1. But here there is a loss of function of P16, which causes inappropriated, unregulated passage to the restriction point in G1 that will lead to cancer and any kind of serious illness. So there will be an overexpression of cyclin D1 when there will be a loss of P16. So this statement is the right statement, because if P16 will not be there, so cyclin D1 will be overexpressed. So again, this statement is the correct statement, and again the right option is option number 3. Now let's come to the next question, that is question number 9 in CSIR unit 4. So there are many superfamilies of adhesion proteins, okay, which play a central role in cell-to-cell adhesion in animals. Ig superfamily proteins are one such adhesion protein. So they are talking about Ig superfamily. They are one such adhesion protein. Which one of the following statement about Ig superfamily proteins is incorrect? So here you have to select the incorrect statement about Ig superfamily proteins. The white blood cell proteins recognized by endothelial cell integrins are called ICAM or VCAM. So we know that endothelial cell integrins, they are used to identify the white blood cell proteins, and these proteins are called as integrins are called as ICAM and VCAM. So these integrins are only known to identify the white blood cell protein. So this option is the right answer. And here you have to select what is incorrect statement. So this option is true, because all of these integrins ICAM and VCAM, that is ICAM, VCAM, they are used to identify the white blood cells protein. So this option is the right answer. Second, these are called Ig superfamily because they contain one or more extracellular Ig-like domains that are characteristics of antibody molecule. So here if we see, this is a correct statement, because just now we have seen it is belonging to the class of Ig superfamily, and they contain more or one or more extracellular Ig-like domains that are characteristic of antibody molecules. So yes, this statement is the right statement. ICAM and VCAM mediate heterotrophic binding to integrate. Yes, whenever this integrin is binding to ICAM and VCAM molecules, always there will be a heterotrophic interaction. What is called as heterotrophic interaction? When two different molecules, or you can say two different cells, communicate with the help of two different molecules that are present over the surface. So if integrin is present on one cell and ICAM and VCAM is present on another cell, so this is called as heterotrophic interaction, because two different molecules are interacting here. Whereas NCAM, they mediate homotrophic binding. Why? Because NCAM in one cell, it is interacting with NCAM of another cell. So this NCAM mediates through homotrophic binding, but ICAM and VCAM, they mediate through heterotrophic binding. They contain large amount of sialic acid, which inhibit adhesion by charge-based repulsion. Now, if you see the structure of Ig superfamily, or you can say ICAM, VCAM, or any of NCAM, you will see they have sialic acid. So you can see the triangle is for sialic acid. This structure is for sialic acid, the purple one. So these sialic acid, they are present in more amount in structure of these ICAM, VCAM, and NCAM, and these are negatively charged. So when they will be negatively charged, they will cause a repulsion. Yes, more negative charge will be there, they will cause a repulsion. But these negative charge, they are going to inhibit the adhesion between cells. And if the adhesion will be inhibited, how it will contribute to the fine tuning of cell to cell adhesion? Because just now we have seen it is going to inhibit the interaction between cells, because all of these CAM molecule, they have more amount of sialic acid. And if it is going to inhibit the adhesion, how it is going to contribute the fine tuning of cell to cell adhesion? So both of these terms are contradictory. Either you have to select inhibit or fine tuning. So it is going to inhibit the adhesion but not the fine tuning. So if it is inhibiting, no fine tuning will be there. So this option is the incorrect answer, or you can say statement. So according to the question, we have to select the incorrect option, and with this D option is the right answer. Now coming to the last question, that is about interferons. So let's see what they're asking about interferons, and here we have to select the combination of statement, we have to read, and we have to select the incorrect option. So interferons alpha, beta and gamma are cytokines. So yes, they are cytokine. Alpha, beta and gamma. Alpha are produced by different types of leukocyte cell, beta is produced by fibroblast cell, and gamma is produced by infected cell. So are cytokine that are produced and secreted by animal cell, very true, after infection by viruses. So when these all interferons are produced, when the cell is infected by any kind of virus. Which one of the following statement about interferon is incorrect? So you have to make sure that you are reading the question correctly, and it is asking about incorrect option. So let's read the first statement. When mammalian cells are incubated with different interferons, yes. So activation of a STAT links stimulation of cell surface receptor with gene expression. So now they are focusing on cell signaling pathway, and we all know that whenever the interferons will be produced by the cell, they will always be recognized and identified by a receptor that is present over the surface of cell, and the name of this receptor is related to non-receptor tyrosine kinase, and this is known to activate the JAK protein and ultimately the STAT protein. So here that's why it is mentioned it is going to activate the STAT dimer. Yes, the activation of STAT will take place with the help of the interferon that is bound to a receptor. So this statement is the correct statement because we have to select the incorrect. So now let's read the next statement, that is option B. Increase in the expression of genes. So whenever there will be expression of genes, so whatever genes will be expressed that are specific to interferon. So once the interferon will bind to the receptor, it will always lead to the expression of genes that are specific to interferon as well. After the addition of interferon alpha results only after the activation of interferon stimulated response element. So what actually happens in cell signaling pathway, as the protein binds to the receptor, there will be an activation of some sort of intermediate protein that is going to bind to the DNA region, and this DNA region is specific for all different types of protein. So here the first protein that is initiating the signaling pathway is interferon. So the interferon is inducing the intermediate protein that is going to bind the specific DNA response element that is called as interferon stimulated response element. So this option is the right answer. The receptors of all three interferons belongs to Ig superfamily of receptor and do not lead to downstream phosphorylation events. All three interferons means they are talking about interferon alpha, beta and gamma. They belong to the Ig superfamily of receptor. They do not lead to downstream phosphorylation event. Just now we have seen it is leading to downstream phosphorylation event, or it is leading to downstream signaling pathway because it is forming one intermediate protein that is about to bind to the interferon stimulated response element, and after this the response of interferon is completed. So it will lead to the downstream events. So accordingly, this option is incorrect as it is talking about Ig superfamily, and just now we have seen the receptors for this interferon, they belongs to the non-receptor tyrosine kinase, not Ig superfamily. So this option is the incorrect one. So accordingly, option 3 goes right. The fourth statement is talking about an important feature of the interferon STAT signaling pathway is its specificity. Each type of interferon induces transcription of a unique subset of genes. Now there are three different classes of interferon. Interferon 1, 2 as well as 3. This interferon 1 class includes alpha and beta. Second includes gamma and third includes lambda. So if you see interferon 1 and interferon 3, they are going to induce some sort of, you can say, the similar proteins. But if you talk about interferon second class, that includes interferon gamma. So they are used to induce some distinct set of genes. So an important feature of the interferon STAT signaling pathway is its specificity, because two different proteins are formed. Type 1 and type 3, they are forming the same protein, some same protein, similar proteins, but type 2 interferon, which includes interferon gamma, that are forming some different proteins. So means different protein expression is taking place with the help of type 2 interferon family. Each type of interferon induces the transcription of unique subset of genes. So again, this statement is the correct statement. So ultimately, we have seen the option 3, that is option C, is the incorrect one. So accordingly, the right answer is option 3. So this is the explanation for the same. So the same that I have explained here is mentioned here too. So now I have come to the end of my session. So I hope the explanation that I have given you for CSIR Unit 4, either it is part B or part C, is beneficial. And if you find the information is beneficial and the explanation is satisfactory, do like and subscribe to the channel, that is Biotecnika. So thank you for your time and patience. Meet you in the next video. Till then, bye-bye. Take care. Have a nice day. Keep learning.